As Alec told you, I'm a scientist, but today I will tell you more about business and how I developed Genos from a startup into a global leader in less than a decade. So Genos was established 14 years ago, is growing organically without any external investment, and is getting bigger and bigger and bigger, but maybe better than this chart is just to look at the images of people working in Genos. There is more and more of us every year, and actually even the last photo is a little bit late. We have even more people today. Today, there are 43 scientists who work in Genos, and I'm particularly proud that many of them got their PhD while they were working in Genos. So they came to me after university, did their PhD studies in Genos, and now they stayed to work in Genos as a PhD researchers. And not only from Croatia, we had researchers or have researchers from 14 different countries in Croatia working in Genos. And we are reasonably successful. We got the award as the most innovative small and medium enterprise in Croatia. We got even the special charter of the Republic of Croatia awarded by the President of Croatia for commercialization of research results. And last year, we were the finalist in the EY Entrepreneur of the Year competition. We were not the winner, but we'll try again. Also, we are the formal member of one of the 10 research centers of scientific excellence. I'm leading the Research Center of Scientific Excellence for Personalised Healthcare. We are recognized not only in Croatia, but also in the world. For example, the scientists ranked us as the best place to work in the world for researchers in industry. For example, Genos was ranked first, while Genentech, which is a Google of biotech industry, was ranked sixth. Vice President of the European Commission, Jyrki Katainen of the last commission, came to visit us in Genos, and this is him talking in Genos, saying that we are the prime example of the type of business the investment fund for Europe aims to support. And now, when leading journalists are telling something about the high-throughput glycomics, they list our small company from Croatia as one of the leading labs in the world in this topic. So what do? We work in the field called precision medicine, and we are trying to discover biomarkers which will enable physicians to tell why people are different. Because today, when you go to a hospital, you are treated as a Homo sapiens. Everybody's equal, we all get the same therapy, and the consequence is that approximately 80% of these therapies do not work. And then you get a second drug, and then you get a third drug, and eventually we find a drug which works. Idea of precision medicine or personalized medicine is that we have a panel of biomarkers. We do the test before the drug is given, and then by stratifying people, you immediately get the right therapy. Everything goes much faster, less side effects, and less expensive. There are huge efforts globally to develop biomarkers for personalized medicine. And the huge effort was directed toward genes and analyzing DNA. But despite technical success, and now it is so easy to sequence genes. It's only a few €100 and you can get every single letter in your genome sequenced. There is very little translation. And actually the expectation of general public, but also the science, was not met with this huge endeavor. And one of the reasons why genomics failed is that genes do not define everything. Yes, many things are defined by our genes, but not that many. Actually, only approximately 20% of longevity and 30% of complex diseases is genetic. Everything else is environmental. And the best example for that are identical twins. On this slide, you can see two pairs of identical twins. Every single letter in their genome is identical, but their life destinies are clearly very different. And we focus on one set of molecules which are making people different and are not directly encoded in our genome, and these are the glycans. So glycans are one of the 4 key components which build life, and practically every protein which appeared in evolution after the appearance of multicellular life is glycosylated. So in this figure, you see a gray part, this is a polypeptide, and the colored ones, these are the glycans. So proteins do most of the work. They are kind of robots which perform all functions on molecular level. But glycans are something which extends the function of this protein. For example, if we imagine a car, protein would be a plate, a wheel, and a motor. If you want to have a windows, rear view mirrors, lights, anything fancier than just the basic thing, you need glycans. One example I like to show are these two birds on this figure. If we look at the bird at the left, this bird doesn't have a single feather, but it's still a bird. So you can study this bird, you can study the physiology of this bird, you can study this bird walking, but you cannot study this bird flying. You cannot study it flying because it does not have any feathers, and the bird without feathers cannot fly. The same thing is valid for proteins. So on the image below, you see the S-glycoprotein, the famous spike glycoprotein of the SARS-CoV-2 virus. On the left, it's a polypeptide part, and on the right is polypeptide plus glycans. And the S-glycoprotein does not work without glycans. Any other protein, they don't work without glycans. And this is not me claiming that. There is a very strong policy document for nearly a decade ago by the US National Academies, which said glycans are directly involved in the pathophysiology of every major disease. And actually, NIH, through its Office for Strategic Coordination, launched the Common Fund program to develop glycoanalytical methods in the US. But this is difficult. Glycans are chemically complicated, they are nonlinear. It's not easy to study them. You have to do many steps to be able to do high-throughput glycomics, and it will take time for other laboratories to develop these technologies. We started 10 years ago and invested over 15 million euros so far to develop this technology, and we did it mainly through different European research projects. And today we are in a position that we already analyzed over 150,000 people, but not any people. We analyze people from the best phenotype and genotype cohorts all around the world. We collaborate with research from Harvard, from Oxford, from Yale, from Imperial, most leading research universities, which enabled us to push glycomics into the mainstream science. And nowadays, it is possible to publish glycomics paper even in leading journals. These are the papers we published in the last couple of years, because we actually finished the initial fishing expedition. So fishing expedition is when you accumulate a huge amount of data and you have no ideas what you're going to catch. For us, this fishing expedition resulted in this huge pile of Excel sheets, which we use now to analyze and to mine the gold out of these glycomic datasets. And the first gold we have, first application of the research we did in the last 10 or more years, is the glycan age test of biological age. For this test, which was developed in Ghana, so by the people on this image, we developed a spin-out called glycan age, which is now already selling this test commercially. Because the idea is, can we reverse aging? Do we have to age? My friend David Sinclair published a book which claims that we do not have to age, that aging can be treated as any chronic disease, and that we can probably prolong life for at least 2 or 3 more decades relatively recently. And in some lab models, this works. There are some pharmacotherapies which make mice A younger than the mice B. If you look at these 2 mice, they're born on the same day, but the one on the left looks much younger than the one on the right, especially if you put them, if you challenge them and you force them to exercise. The one on the left runs like crazy, while the one on the right hardly moves. And they're exactly the same chronological age. So the years, the number of times the sun is going, the Earth is going around the sun, is not something what determines our biological future. It's molecules which change as we age. And something which is in the center of aging is inflammation. Actually, there is a theory called inflammaging, saying that inflammation is affecting many different molecular processes, which then result in aging. And when we think about inflammation, it's actually a repair mechanism. When there is infection, when there is a damage, our cells come in, destroy everything, and then rebuild what was wrong. And this is fine if there is infection, and this is fine if there is damage. But if there is excessive inflammation, if there is too much inflammation, and this is happening when we are getting older, then we have something which is called low-grade chronic inflammation, which is actually damaging our body. It's making more and more little scars. It's consuming huge amount of energy. And in this way, it's one of the drivers of aging. And glycans attached to immunoglobulin G are one of the molecules which balance this process. For example, when we are young, we have these glycans on the top, which are suppressing inflammation. So our immunoglobulins are suppressing inflammation, and we have a lot of energy. We don't waste it on inflammation. But as we are getting older, our immunoglobulins start to promote inflammation, and we are entering this vicious circle of inflammation, which is driving aging. And through this glycan age test, we have an objective, quantitative measure which integrates genetic information, so what is written in our genes, epigenetics, our past history as an individual, and current environment. And something you can measure, and something you can actually change. Because most of the biomarkers of aging cannot be changed. Epigenetics cannot be changed easily, telomeres cannot be changed easily, but glycans can. So if you do something which is known to be healthy, for example, you lose extra weight, your glycans improve and they're not so much inflammatory anymore. And this test is something you can easily do. It's a home test. You take it, take a drop of blood, send it to the lab, and get a number with the idea that you actually want to prevent diseases which come with aging. Because normally what we do, we go to hospital when there is pain somewhere. Something hurts, something in our body is not functioning, and then we ask for help. And we expect a magic pill which will solve our problem. And these magic pills are usually made in a way that you have to take it every day for the rest of your life. And then you have something called chronic disease. And usually you cannot fix it. You are sick for the rest of your life. But if you go backwards and you detect a disease in an early stage, when there is no huge tissue damage, just only biomarkers which cause this damage are changed, then doing maybe even a simple lifestyle intervention, sleeping more, being under less stress, eating a bit less, can prevent this long-term damage, and you will actually go back to homeostasis and not end in a disease. And we know what are the easy things to do, what are the important things to do, but we don't do them because they're not easy. It is not easy to do something today, to be healthy after 30 years. And the problem is, reward comes too late. I'm not going to skip this cookie because I will be healthier in 30 years. No, I'll do it tomorrow. I'll eat the cookie today. But if we would have an objective biomarker, something that will tell us in a few weeks, what have I done in the last few weeks, then people will get more motivated. And we already see this in the real world. There are some clinics, there are some guides who actually help people to change and to improve. And then you see people in the late 70s dancing like this, which is really giving hope that we do not have to age quickly. And when I say age, I don't mean that we have to live sick and old forever, but we should stay young and healthy for a longer time and then eventually get old. And I thank you very much for your attention.